Okay. Now it is my first great pleasure to introduce our invited speaker, Juliana Calábria de Araújo. She graduated in biological sciences at the Federal University of Rio de Janeiro and did a PhD at the University of São Paulo. I just learned she was once at my institution for an internship, so we have some common colleagues, and a postdoc at USP. She has a long history of working on wastewater treatment and anaerobic treatment microbiology. She's now an associate professor in wastewater microbiology at the Department of Sanitary and Environmental Engineering at the Federal University of Minas Gerais in Brazil. She will present her work on the surveillance of antimicrobial resistance in Brazil, as revealed by long-term monitoring of wastewater treatment plants. We're looking forward to your talk, Juliana.
Okay, thank you so much. So, thank you, Helmut and Sarah, for the nice introduction. Thank you all for coming here today to listen to this talk. And it's a pleasure and an honor to be here today. So I also would like to thank the organizer of EDAR7, especially Professor Dominique Frigon, to give me the opportunity to show the results of our research. So I'm going to present results from different studies that we have been conducting in the past 8 years, measuring antibiotic-resistant genes and antibiotic-resistant bacteria in different wastewater treatment plants in Brazil, but specifically in the area that I live, which is the Belo Horizonte, which is the capital of Minas Gerais state. And I must say that this is the name. Minas means the mining mining state. So Belo Horizonte is located in the Iron Quadrangle area of Brazil, where we extract iron and gold and other types of metals. Okay, so you all know this figure, so I don't have to say it again, that we are facing a very huge public health problem due to antimicrobial resistance. And this has already been shown that by 2050, we will have about 10 million deaths due to antimicrobial resistance. But in Brazil, the toll is also high, of course, comparing our population. So a study pointed out that in 2019, we had about 138,000 deaths attributed to antimicrobial resistance. So it's also a big concern for us. And especially, well, due to different factors. One of them is related that Brazil is one of the top 5 countries that consumes lots of antibiotic in livestock production. And as you can see in this figure, by 2030, Brazil would be the 3rd largest antibiotic consumer in livestock production. And here I'm showing the antibiotic consumption in Brazil by package that has been sold from 2014 to 2020 to point out the top 5 most consumed antibiotics in Brazil, which are amoxicillin, azithromycin, cephalexin, ciprofloxacin, and levofloxacin. And I'm going to show you that, for instance, quinolone-resistant genes are one of the most, one of the most higher genes detect in wastewater treatment plants. Okay, and also, well, I saw this picture today 2 times in other presentations, so you might also be familiar with this paper of Hendrickson and collaborators from 2019, showing that the antibiotic resistance diversity and abundance varied a lot between countries. And as you can see here, Brazil is colored dark blue, which means that there is a high abundance, but this was predicted by very few data from this area. So it's very important for us that we can generate our own numbers and data. And of course, with this information, take action to minimize and prevent the dissemination of antibiotic resistance into the environment. Okay, so how about the sanitation condition in Brazil? As you can see here, we have data from 2021 from the Brazilian National System for Water and Sanitation. And here, you can see in relation to potable water, we have more than 93% of the urban population in Brazil have access to potable water. So this is, we are almost reaching the universalization of this type of sanitation service. But regarding sewage collection, the numbers are not good. So as you can see, considering the total Brazilian population, we have only 53% that had access to sewage collection and then sewage treatment. When we look to the urban population, this number increased a little bit. So 64% of our urban population had their sewage collected and then treated. But the total number of the treated sewage in Brazil is about 51%. It means that from all the sewage produced, the flow of the raw sewage produced, we only treat 51%. So it's very, it's not a good number. And this, for all the problems that this can cause in the environment, especially also thinking about the antimicrobial resistance dissemination by the sewage. Okay, so here is the map of Brazil. And as you can see, the Minas Gerais is the red point in the southeast part. And then this is a zoom of the Belo Horizonte city. The picture, the first one up, is a picture of the city. So we have, we are surrounded by mountains. And we are about 2.6 million people in Belo Horizonte city. And the city is located in these 2 sewer shed basins. So in the south and center part of the city, we have sewer shed basin A, and the sewage produced here is collected and treated here at the wastewater treatment plant A. So 1.4 million people is living here. And in Belo Horizonte, we have a good sewage coverage. So 94% of the population, the sewage produced by this population is actually collected and treated. And then treated here by this wastewater treatment plant that uses conventional activated sludge system. And in the north and northeast part of the city here, we have 1.3 million people located here, and the sewage produced by this population is treated here at the wastewater treatment plant B, which uses USB reactors. So this is the picture of this onça. We call onça because the river here is the name of the river. So this wastewater treatment plant uses USB reactors, so anaerobic treatment, followed by biological treatment filter to treat the sewage. So what we did was we, over more than 1 year, actually 16 months, we collect the raw sewage and the treated effluent from wastewater treatment plant A, located in sewer shed basin A, and also from the wastewater treatment plant B. So I'm going to show the results of this study. But we also investigate another wastewater treatment plant, which here I am calling C, located in a different municipality, in a very small city, which is called Ibirité. And this is a very interesting wastewater treatment plant because they have a modified activated sludge followed by this lamellar decanter, which they applied some physicochemical treatment, and then the treated effluent goes into a UV reactor to disinfect this effluent. And then the treated effluent goes to the river, but also goes to the Petrobras, which is our national petroleum company. So Petrobras has supported, let's say, this wastewater treatment plant to produce this tertiary effluent to be used back into the industry. And I'm also going to show you some results of this. This is another wastewater treatment plant located at our international airport at Confins, close to Belo Horizonte. So this is another type of wastewater treatment plant that uses maturation pond followed by facultative pond, so only biological treatment. Okay, so the first work is this one. It has already been published, and we monitor the 7, actually, antibiotic-resistant genes in the raw sewage and also in the treated effluent. And then we also did some 16S amplicon sequencing in order to look at the community diversity in these samples. So, as you can see here, so we have the 3 wastewater treatment plant, the A, which uses conventional activated sludge, B, which use anaerobic reactors, and the other, the C, the small one, which has modified activated sludge followed by UV disinfection. So we have some removal, about 0.51 log units by these 3, for these 3 genes, and a higher removal, about 1.5, sometimes 2 unit logs for Blatain, RMB, and KNRB, comparing the 3 wastewater treatment plants. When we look at the microbial community, and here in this graph, I am pointing the top 10 genus that we found at the influent and also effluent of the 3 wastewater treatment plant. And then here you can see at the NMDS that the samples clustered apart, and due to because they are receiving sewage from different population, from different catchment areas. And here, I want to point out some of the genus that we could find at the influent, such as Cloacibacterium, Acinetobacter, Acidovorax, Streptococcus, and these were also found at the treated effluent from the anaerobic wastewater treatment plant. And we also look at the sequence trying to select the genera that are recognized as potentially pathogenic bacteria. And here I am showing the 25 genera that has some species that are really pathogenic. And these sequence represent about 22% of the identified sequence in these samples from raw sewage and also from treated effluent. And I have to point out some genera in raw sewage. Again, Acinetobacter, Archobacter, Streptococcus, and Enterobacter. They were prevalent and abundant in raw sewage. We also did some network analysis correlating the genes that we monitor in raw and treated sewage, and also the genus that the prevalent genus that we could find in these samples. And as you can see, for instance, for the macrolide gene, RMB, this gene shows the highest number of correlation with the pathogenic bacteria, followed by the blatain gene and also KNRB. And we couldn't see, actually we saw a negative correlation between gram-negative bacteria and the genes that we investigated in this study. So again, I'm going to show the same 3 wastewater treatment plant that we monitor, but now in this study, we quantify antibiotic-resistant bacteria by cultivating and isolating these bacteria from raw sewage and also from the treated effluent from all the 3 plants. And after isolation, we identify more than 200 colonies, and then we tested these colonies using antibiogram to see the phenotypic characteristics of these isolates. So first, I'm going to showing in this table the removal of these antibiotic-resistant bacteria. So we isolate these bacteria against 11 antibiotics, such as ampicillin, chloramphenicol, sulfamethoxazole, amoxicillin, ciprofloxacin, and so on. So as you can see, for wastewater treatment plant A that uses conventional activated sludge, the average removal of these resistant bacteria was about 1 log, 1 unit. For the wastewater treatment plant B, the removal was very bad, about 0.4 logs. And for wastewater treatment plant C, that uses the modified activated sludge, followed by physical-chemical treatment, and then UV disinfection, the removal was from 2 to 3 logs. But this is interesting, and it is interesting to point out that after UV disinfection, we didn't see any removal. So the removal for this wastewater treatment plant C was occurring by the biological treatment, by the sedimentation part, and then by the physical-chemical treatment applied at the lamellar decanter. So here, okay, you are not supposed to be able to read this, but from the isolates we tested, we did antibiogram test, and then we could find that most of the isolates, as you can see here, about 74% of the isolates showed multi-drug resistance, so resistance to more than 3 different classes of antibiotics that we have tested here. And at the upper part of this table, we have the antibiotics that were tested, such as ampicillin, amoxicillin, 3 different carbapenems, such as meropenem, imipenem, ertapenem, sulfamethoxazole, and so on. And what is important to be aware of is that 6.4% of the isolates that we recovered from the treated effluent showed multi-drug resistance. So still, If we are reducing the concentration of the antibiotic-resistant bacteria, this is good, but it's still the ones that are being released and discharged in the environment are showing multi-drug-resistant phenotype. And among them, 52% were resistant to the last resort, one of the last resort antibiotics, such as meropenem, which is a carbapenem. Okay, so in another study, we investigated the wastewater treatment plant C, the one that uses a modified activated sludge followed by the physical chemical treatment, and that uses UV irradiation as disinfection step. And in this study, we did a metagenomic analysis. So you can see APT means after preliminary treatment, which is the raw sewage. So this is the microbial community in raw sewage after the biological treatment, and the third column is after UV disinfection. So we can see, for instance, for Proteobacteria, mainly gamma Proteobacteria, there was an increase after the treatment. But for Firmicutes, for instance, there was a reduction in the relative frequency along the treatment. When we analyzed the genus, so here I'm showing the top 10 genus identified. Again, we can see a reduction, for instance, in the abundance for the Acinetobacter after the biological treatment, but then we saw a slight increase in the abundance of Acinetobacter, forgive me, for Acinetobacter after the UV disinfection step. And here, again, I'm only showing what is going on for Acinetobacter and Streptococcus during the treatment. So for Streptococcus, for instance, we can see significantly reduction in the concentration of Streptococcus during the treatment, after biological treatment and also after UV disinfection. And here in this principal component analysis, we can see that the samples were separated, but not by the treatment, but by the month of collection. So here we have the raw sewage, and then the biological treated and the UV disinfected samples. Okay, and here, so we have some of the metagenomic results. In the, here you have the 20 most abundant genes. And for the first 4, we can see that there was no reduction in the frequency along the treatment. And in this other graph, we saw the most abundant antibiotic-resistant genes in plasmids. And I want to draw your attention for the MPHE and MSRE, which are genes that confer resistance to macrolides that increased after the treatment, so after the UV disinfection. Okay, so here we have some results for the 4th wastewater treatment plant. So it's the wastewater treatment plant at the airport, at the international airport in Belo Horizonte. So for 10 months, we collect samples from the influent and also from the treated effluent and quantified antibiotic-resistant bacteria and also antibiotic-resistant genes. And this is very interesting because we can see a very high removal, about 2 to 3 log units for both for antibiotic-resistant bacteria and also antibiotic-resistant genes. So this could be due to the fact that the ponds has a very high hydraulic detention time. It's about 4 to 5 days. Well, I know that you must know what is hydraulic detention time. So this helps the bacteria to settle, and it was important for the reduction of the concentration of these two pollutants. And when we identified the antibiotic-resistant bacteria in the raw sewage and also in the treated effluent, we also found that some species were showing mold drug resistance phenotype, mainly at the E. coli, the Escherichia genus, and also at the Enterobacter genus. So still, we were able, or this wastewater treatment plant were able to reduce by 99.9% of the concentration, but it's still the bacteria that were present in the treated effluent showed mold drug resistant phenotype. And this is another study, and you can know more by stopping by and seeing the posters 14 and 19, which is the master work of Ana Paula. And so in this study, we monitored the same two full-scale wastewater treatment plant of Belo Horizonte, but looking, quantifying now 12 different types of antibiotic-resistant genes. As you can see here, most of the genes were reduced by 1 to 1.5 logs, but the TET-X genes were not reduced, were not removed at all. Actually, in the treated effluent, the concentration increased compared to the influent. So, meaning that maybe this wastewater treatment plant was acting as a bioreactor to increase this type of resistance. And for the other wastewater treatment plant, we only saw removal for these 3 genes, but a very slight removal, about 0.5 logs. And when we calculated the antibiotic resistance load of this wastewater treatment plant by each genes, as you can see here, the numbers are quite the same, despite the removal efficiency was completely different. So meaning, again, that although we have been treating the sewage, the load of the genes that is being discharged is very high. And okay, to sum up, so we saw that the removal of antibiotic-resistant bacteria and antibiotic-resistant genes depends on the biological treatment that the wastewater are applying in the process. We saw some reductions for some genes. For instance, for SU1 and TET-A, the reduction was about 50% compared to the other genes. Blatain, ARMB, and KNR-B, that we saw a much higher removal for the 3 wastewater treatment plant that we investigated. And the wastewater treatment plant located at the international airport removed more. The removal efficiency was higher compared to the other 3. Yeah. And all this work, the conclusion that we can take from this is that we need to improve our sewage collection and treatment in Brazil. And although we have some treatment, still we are releasing high loads of antibiotic-resistant bacteria and genes in the environment. So that said, I'm going to thank you for your attention and all the students that have been doing this work, and all the Brazilian agencies that have been funding these students. Thank you so much.
Hi, I'm Stephen Georgievic from University of Technology in Sydney. I was wondering if you've sequenced the E. coli organisms that have survived your wastewater treatment plants that still show high resistance to carbapenems.
Not yet. Yeah, this is it's a thought for a future work, but we still have the isolates frozen at -80. Yeah.
Okay, thank you.
If there's another question.
Well, Jan Siemens from Gießen here in the very back.
Okay.
I found your results with this pond system very promising and interesting. So would you recommend these kinds of stabilization ponds or treatment ponds for more decentralized wastewater treatment in, let's say, rural areas? Because it's much, much cheaper than to have a centralized system with all the infrastructure.
Yeah. You are right. So the results are really very promising, and we have to think that the flow that this wastewater treatment plant treats, it's for a population equivalent and 25,000 inhabitants. So for a small village or a small community would be recommended. Yeah, but we also have to take into account about the climate of the region that you want to apply this type of biological treatment, because in Brazil we have so in Belo Horizonte, the average temperature all over the year is about 24 degrees, which is very warm. But in other parts of the world, this could not be a feasible, let's say, biological treatment due to the temperature. But yeah, if you have temperature, good temperature and area, this is a very good biological treatment.
Okay, I'd like to welcome our next speaker, Muhammad Adnan Sabar. Please come
Very good afternoon, everyone. And thank you very much for coming here for us to. And my name is Muhammad Adnan Sabar. And right now I'm a postdoctoral fellow in Chulalongkorn University in Thailand. And the topic of my presentation is intercontinental diversity of antimicrobial rhizostome and mobilome in activated sludge. So as you know, in the environmental domain, the aquatic environment, particularly the wastewater, is the most important source for the dissemination of the antimicrobial resistance. And all of the domain, including the humans, animals, aquaculture, and the fisheries, they are utilizing the antibiotics and their byproducts, including the waste and sometimes the mishandling they are releasing into the wastewater. And also the effluent of the industry, they are also heading towards the wastewater treatment plant. So the wastewater are the important environmental reservoir of the antimicrobial resistance. So in that case, adopting the One Health approach is imperative, as the resistance bacteria and the resistance genes can move into any of the domain in any site. So as you already seen many times, so the recent past studies it has been investigated the global diversity, commonality, and the landscape of the antimicrobial resistance in the untreated wastewater, and which showed the linkages with the socioeconomics, health, and the environmental factors. And in our recent study, it showed that the antimicrobial rhizostome in the activated sludge is different as compared to the untreated wastewater. This might be because of some of the factors, including the microbial community and the mobile genetic element, which found different in the activated sludge as compared to the untreated wastewater. So as the effluent of the wastewater treatment plant is also impacted with the rhizostome of the activated sludge, and which are discharged into the environmental water. So despite importance of the activated sludge as an important environmental reservoir of the AMR, so this remains unexplored. So the objective of this study is to understand the international diversity of rhizostome in activated sludge, and also to reveal the ARGs which is unique and characterizing the country. This could be the useful knowledge for understanding the determinative factors of the rhizostome in activated sludge. So for that, we acquired the metagenomic data set from different countries, including the Japan and China in the case of the Asian continent. Also, specifically the Denmark in the case of the Europe, and the Eastern and the Western USA. In the case of the Japan, we acquired the metagenomic data set from our last study, in which we collect the samples of the wastewater and the sludge from 5 different wastewater treatment plants, which were located at different regions in the Japan. And we collected the sample during the winter and the summer season. And the library was prepared by using the shotgun metagenomic sequences. Regarding for the other countries, the metagenomic data sets was acquired from the databases, including the DNA Data Bank of Japan and the ENA, by having the search keywords of the metagenomics of the wastewater and the sludge and their selection criteria you can see here. So overall, we obtained 39 different data sets, metagenomic data sets in the case of the activated sludge, including the Japan, Eastern and the Western of the USA, China, and the Denmark. And we acquired 23 data sets of the wastewater, having from the Japan, Eastern, and the Western of the USA, and the EU, especially the Denmark, Sweden, Switzerland, and the Spain. So all of these metagenomic data set was utilized to conduct the metagenomic analysis by using the NIG supercomputer. And the flow of this analysis you can see here. In general, the raw sequences was first trimmed and the quality of the read was maintained. And later on, the quality reads without merged parents were utilized to do the blast. And we conducted the blasted by using the card database in the case of the ARGs, while we use the mobile genetic element database in the case of the MGE analysis. And later on, the profiling of the ARGs and MGEs of each sample of the activated sludge and the wastewater was made by having the gene family, gene name, drug class, mechanism, and so on. And we also calculated the count of the 16S bacterial read by the Silva database. To make the profiling in each of the sample, we conduct the normalization. In the case of to normalize by the gene length, we acquired the read per kilo base pair by getting the read counts of the fragment by the length of the gene in the base pair. We also calculated the ARG composition, which is actually the proportion of each ARG in the entire rhizostome of the sample. By calculating the RPK of the specific ERG in a group divided by the entire ARG in a sample. And we also calculated the total ARG abundance by utilizing the entire ARG copies in terms of the RPK by using the entire bacterial population based on the 16S microbial. So here we got some of the important results which showed that the abundance of the total ARG abundance based on the 16S microbial community is lower and the less diverse in the case of the activated sludge as compared to the wastewater. And we also found the ARG abundance consistent in some of the countries, including the Denmark, eastern of the USA, and Japan in the case of the winter. However, we also found some, like the diverse ARG abundance in the case of the Japan during the summer, and the western of the USA and China, which depends on the location as well. And similar trend we found in the case of the total MGE abundance, which showed the lower and the less consistent abundance in the case of the activated sludge as compared to the wastewater. And this highlighting that the mobile genetic elements are intentionally play an intrinsic role in the case, a role in the shaping of the rhizostome universally. So here we found some of the core ARG abundance which varies between the activated sludge and the wastewater. And these 11 core ARGs constitute majority in the case of the activated sludge in each of the country, which are making up to the 80% of the rhizostome in activated sludge by comparison with the wastewater, which are showing less than 30%. And these core, which highlighting that these core ARG are persistent and enriched inside of the wastewater treatment system. Specifically, the SAL1 and the QUEC E delta we found like the prevalent in the activated sludge globally, except for the Western USA. And in the case of the ESBL gene, the OXA were found prevalent in both of the case of the activated sludge and the wastewater. So here you can see the principal component analysis which were conducted by utilizing the ARG abundance, ARG composition, both in the activated sludge and the wastewater, which are showing that the rhizostome is primarily defined by the activated sludge and wastewater instead of the country. And for to check the intercontinental diversity of the rhizostome in activated sludge, we only conducted the PCA analysis by using only in the sludge, which showed that the Japan were characterized by the positive PC1, while the Denmark and the Eastern USA was characterized by the positive PC2, while the China and the Western USA were characterized by the negative PC2. Overall, it showed that the rhizostome in the sludge were less diverse as compared to the wastewater. So here we also here you can see the relation of the ARGs belong to the different drug classes with the microbial community that we found both in the activated sludge and the wastewater. And we found 4 different microbial groups. The microbial group A mostly contain the phylogenetic classes of the anaerobic bacteria, which were found abundant in the untreated wastewater. And they showed the high correlation with the ARGs belong to the clinically important drugs, including the quinolone, macrolide, tetracycline, and the beta-lactam. While in the case of the group D, it mostly contained the phylogenetic classes of the aerobic heterotrophs that were found abundant in the activated sludge. And they showed the high correlation with the ARGs belongs to the multi-drug by Eflex, sulfonamide, and some of the ARGs of the macrolide and the aminoglucoside. And the core ARGs also showed the high correlation with the group D. And similar trend we found in the case of the group C, which have the phylogenetic classes of the marine and the soil prokaryotes. So here you can see the relationship of the ARGs with association with the mobile genetic elements. So the regression analysis showed the high correlation of the MGEs with the total abundance of the ARGs, both in the case of the activated sludge and the wastewater. This highlighting that the mobile genetic element are facilitating the ARG dissemination in both in activated sludge and the wastewater. And here and also we found that in the case of the mobile genetic element, specifically the INT1 and the IS91 was found abundant in the activated sludge with comparison to the wastewater. And they showed the high correlation. Most of the ARGs belong to the core ARGs. So it highlighting that the activated sludge facilitated the mobility of the core ARGs via the mobile genetic element, including the INT1 among the microbiomes. So here you can see the conclusion. So the antibiotic rhizostome in the wastewater and the sludge is primarily defined by the sludge and the wastewater rather by the country. And the core ARGs comprise nearly the half of the total ARG abundance in the activated sludge, that promoting the intercontinental uniformity. And the core ARGs are innately incorporated in the mobile genetic elements of the sludge microbiome and selectively reserved in the activated sludge. Thank you very much for being me.
I was wondering, so you saw this difference between the main genes and the hosts in the wastewater and the sludge. For the genes that were in the sludge, could you determine if they were also present in the wastewater, although not the dominant ones? Or it's a totally different community and these genes kind of arose in the activated sludge and were not present in the wastewater?
Yeah, so the abundance of and the variety of the ARGs were higher in the case of the untreated wastewater. And these ARGs were also found in the untreated wastewater. But in the case of the core ARGs, their abundance was higher in activated sludge without the discrimination of the country. So there was some of the differences among the abundance.
Hey, next I would like to welcome our next speaker, Claire Gibson from the lab of our host, and she will speak about the complex fate of antimicrobial resistance genes in activated sludge determined by solids retention time.
Hi all, thank you for being here today. So I'm sure I don't need to tell anyone in this room that the environment plays a critical role in the emergence and spread of antimicrobial resistance worldwide. Wastewater treatment plants have been identified as a particularly important hotspot of antimicrobial resistance because it's been shown that treatment technologies don't achieve complete removal of antimicrobial resistant genes and bacteria before they're released into the environment. And this is through both the effluent wastewater flow and the waste biosolids produced during the wastewater treatment process. So it's really critical that we get a better understanding of exactly what is impacting the persistence of antimicrobial resistance determinants within wastewater treatment plants to allow us to develop new mitigation strategies to minimize the release. So for all of you who are not necessarily familiar with wastewater treatment plants, here I'm showing you a basic schematic of the activated sludge wastewater treatment plant. Now, as the name suggests, it's a biological process, meaning it relies on the activity of microorganisms to break down the contaminants within the incoming wastewater. So upon entry into the wastewater treatment plant, our influent wastewater is combined with this rich and diverse microbial community which is present within the activated sludge. It then enters into an aeration tank where oxygen is provided for the metabolism of the bacteria and the breakdown of contaminants. This then enters into a settling tank, where by gravity settling, we separate out the solid and liquid portions, with the clean effluent wastewater discharged into the environment, and the solids either recycled within the system or disposed of. Now, as you can probably imagine, there are a number of different variables that could impact antimicrobial resistance within these systems at any one time. So it's very difficult to pinpoint the relative contribution of each. But a recent study using highly controlled reactors has found that influent immigration plays a key role in shaping the microbial community composition within the activated sludge, and it also causes a significant increase in the abundance and diversity of antimicrobial resistance genes. Now, these changes to the antimicrobial resistance genes in the treatment plant were attributed not only to direct immigration of ARGs, so the movement between the sewers and the activated sludge, but also the microbial community changes which are caused by this influent immigration process. Which brings us on to the question, what factors are actually impacting this influent immigration process itself? So in the current study, we wanted to investigate the impact of wastewater treatment plant operation on this influent immigration process, with a specific focus on the impact of solids retention time, or SRT. So for all of you who aren't necessarily familiar with wastewater treatment plants and what solids retention time is, it's essentially the mean amount of time that biosolids are retained within the wastewater treatment plant. So if we come back to our very basic schematic of the wastewater treatment plant, the SRT is dictated essentially by the amount of solids which are recycled in the system through the return activated sludge and the amount that are disposed of. So if we have a higher amount of solids which are being recycled within the system, they're obviously going to be maintained within the wastewater treatment plant for a longer amount of time, meaning we have a higher solids retention time. So as I mentioned earlier, there are multiple different variables impacting the microbial community and antimicrobial resistance at any one time within these systems. So it would be very, very challenging, if not impossible, to study the impact of SRT with appropriate reproducibility and controls in a full-scale wastewater treatment plant. So instead, we chose to use a series of highly controlled reactors to investigate this. So all of our reactors were inoculated with activated sludge taken from a full-scale wastewater treatment plant to help us to try and have a microbial community as close to what we would find within a full-scale wastewater treatment plant. And we operated all of these reactors at different SRTs, so between 1.8 and 14 days SRT. And all of them were fed with a synthetic wastewater to allow us to keep the wastewater composition stable over time. Now, the reactors were operated in two different phases. So during phase 1, all reactors received synthetic wastewater only. Now, the main purpose of this was to establish a stable, steady-state community at each SRT before we introduced immigration during phase 2. Now, we introduced immigration by supplementing our synthetic wastewater with influent solids, which were harvested from a full-scale wastewater treatment plant again. And these were then supplemented to the synthetic wastewater feed and added into the reactors. We also included a sterile control, which included sterilized influent solids. So they were sterilized using gamma irradiation, which was supplemented into the synthetic feed as well. And the reason for this was to see whether any impact we were seeing with immigration was due to actively growing immigrants or just because we were adding an additional food source into the system. So, quick reminder, phase 1, the main goal was just to establish a stable, steady-state microbial community, and it was during phase 2 that we were testing the impact of immigration. So, let's begin by taking a quick look at what happened during phase 1. So, just a quick reminder, all of our reactors were inoculated with the same microbial community, which originated from activated sludge of a full-scale wastewater treatment plant. But despite this, at the end of phase 1, we found unique microbial communities were formed based upon the SRT of operation. So you can see on this PCOA here that the higher SRTs clustered towards the left and the lower SRTs clustered towards the right. Now, when we introduced immigration during phase 2, let's take a look at what happened. So I know there's a lot of different points on this PCOA, so don't be too overwhelmed. But essentially what you're seeing at the bottom here in the circular points is what we saw on the previous slide, so at the end of phase 1. And after immigration, the points are shown in triangles towards the top of the PCOA. And essentially what we saw was that the SRT effect was maintained, even when we introduced immigration into the system. But we also saw a clear immigration effect too, with all of the microbial communities becoming more similar to that of the influent solids. So obviously, we can see visually here that SRT is having immigration is having an impact regardless of the SRT of operation, but we wanted to be able to quantify this effect and look at specifically what was immigrating within the systems. So to do this, we developed a series of population definitions to break down the community into different groups. So firstly, we defined the core resident populations. So these were taxa which were present in the reactors regardless of whether there was immigration or not. So firstly, we had our universal core residents present at all SRTs within the microbial community. And then we had our SRT core residents, which were only part of the core resident community at selected SRTs. We then had our immigrant populations, which were only introduced into the system during phase 2 when we introduced immigration. So again, we had universal immigrants, which successfully immigrated at all SRTs, and SRT immigrants, which only successfully immigrated at selected SRTs. Now, we went on to quantify the relative contribution of each of these populations towards the overall microbial community. So I know there's a lot of numbers in this table, but I just want to draw your attention to the immigrant population here exclusively. So what we noticed was that there was actually a positive correlation between reads contributed through immigration and SRT. So at higher SRTs, we were seeing immigration to have a greater impact on the overall microbial community composition. Now, this might not seem that exciting to you, but it actually kind of goes against what other studies have predicted. So in the past, studies have hypothesized that at lower SRTs, there's likely a higher influence of immigration, and we're showing that it's in fact the opposite here. So what we think this might be associated with is that people often think of SRT as a form of disturbance. So they assume that at lower SRTs, we have higher disturbance, which opens up niche for immigrants to colonize. But in fact, if a wastewater treatment plant was operated at lower SRTs for a long period of time, it's likely that the microbial community is quite well adapted to these conditions, as we established during phase 1 by developing our stable, steady-state microbial community. So now we've got this interesting trend with SRT and immigration. Our next question is, how was this impacting antimicrobial resistance? So we could just go in and measure a bunch of genes, but then the risk is that we have a bunch of genes and we don't know what any of it means. So instead, we wanted to begin by hypothesizing what we might see based upon the populations that we've identified so far. So if we look at our immigrant populations in a little more detail, so take, for example, our SRT immigrants, which are shown towards the right in that red box there, you might notice that at higher SRTs, we have a greater number of reads contributed through this group. And this is likely contributing towards this positive correlation that we're seeing in the graph on the right here. So if antimicrobial resistance genes are associated with this population, we might expect to see something like this when we look at the abundance of these genes. Similarly, if we were to look at the universal immigrant population, we might expect something like this, because we see that there's a similar number of reads introduced at all SRTs within this population. And there's also the chance that immigration doesn't have an impact at all, in which case we might expect a similar gene abundance both within our control group and our group with immigration. So we decided to use high-throughput quantitative PCR array through ResistoMap to quantify 95 different targets, including antimicrobial resistance genes, mobile genetic elements, some taxonomic markers, and metal resistance genes. And we used a multiple regression analysis to see whether any of these trends were present within our data set. And overall, we were able to identify 7 different groups linking our population dynamics to antimicrobial resistance. So to show just a few examples based upon what I've already discussed, we found cases where the abundance increased at a similar level at all SRTs, as shown on the left, which suggests that they're associated with the universal immigrant population. In other cases, we saw a significant increase with immigration, but also a positive correlation with SRT, which suggests that they're associated with the SRT immigrant population. Now, if we think about this a bit more practically, thinking back to the wastewater treatment plant, if we were to alter SRT with the hope of reducing antimicrobial resistance genes in the situation shown on the left, it would likely have very little impact on the abundance of these genes. But if we were somehow to introduce a pre-treatment to minimize the level of immigration altogether, it's likely that this would cause a significant decrease in the abundance of these genes. Whereas on the right, by altering the operational parameters alone, for example, by reducing the SRT, we could significantly decrease the abundance of these genes. And of course, there was also situations where immigration did not significantly impact the abundance of genes at all. So take, for example, these 2 groups here. We saw both a positive and negative correlation with SRT, but immigration did not affect the process at all, which means that if we were to target immigration as a means of controlling AMR within these wastewater treatment plants, it would likely have very little effect on these abundance of these genes. So overall from this study, this really shows that ARG dynamics are highly complex. So there really isn't a one-size-fits-all way of managing AMR within wastewater treatment plants. And it's likely that multi-barrier approaches will be required in the future to manage antimicrobial resistance, or we may need to prioritize targets of interest. And with that, I'd just like to thank all past and present members of the MyCell lab at McGill University, the operators at the wastewater treatment plant where we sampled, and the funding organizations as well.
Andrew Singer, UKCEH. Really interesting. We missed the source of the immigrants in your study. What was it? Untreated sewage?
So we used a synthetic wastewater as kind of the feed, but we took the actual solids from the full-scale wastewater treatment plant. So it was a matter of separating at the treatment plant the solids from the wastewater was there, and then it was added into this synthetic feed. So it was full-scale microbial community immigrants, but synthetic background.
And what was the HRT from the sewage works that you got that from?
Ooh, I cannot remember the exact value, but I can get back to you on that.
I'm just, I think it's really curious that you got the result you did, and whether there's a relationship between the result you got and the inoculum that you had. So was the inoculum an inoculum that had already adapted to the less labile chemicals, and so they feasted on the things that were left over after the really labile chemicals were already gone, and whether that could, basically it's just skewed your result based on the fact that you had things that feast on hard to degrade things.
Yeah, it's quite possible. Another thing we also considered is a lot of studies often just compare the shared ASVs between the two environments as an indicator of immigration. So to test this hypothesis, we did that, but at the end of phase 1 to see, the shared even before we've introduced immigration. And we found that at lower SRTs, the community is naturally more similar to that of the influent solids. So even though there was never any immigration, if we were simply using the metric of shared ASVs, you would assume that there was higher immigration. So I don't know whether it's just an experimental flaw in the assumption that shared equals immigrants. That we've been able to tease out because we had this control group included as well. So it's also another possibility.
Our next speaker is Stella Christou.
Hi everyone, I'm Stella Christou and I'm a PhD student in Environmental Engineering at the University of Surrey in the United Kingdom. And my presentation today is on exploring AMR in conventional and advanced wastewater treatment processes. And that's a cross-study along the oxygen gradient. So for those who may not be very familiar with wastewater treatment plant processes, their default role is to receive wastewater discharge, treat it through different stages that might differ between different facilities, and returning to the water bodies or further use it, for example, in agriculture irrigation. So, in short, by treatment, we mean the removal of organic matter, nutrients, pathogens, and other contaminants that are present in the influent. So when we refer to secondary treatment, we mainly refer to biological wastewater treatment that employs microbial communities in order to perform this treatment. And these communities, the indigenous communities, while working towards that aim, they come in contact with other antibiotic-resistant bacteria, antibiotics, antibiotic resistance genes that are present in the influent, and along with other contaminants that may act as co-selection factors, they create the suitable conditions for AMR dissemination. And since wastewater treatment plants' default role is not to treat AMR, there is a possibility of further spread to the environment. So during the past years, with the population growth, increased pollutant load discharge, and increased wastewater generation, there is a need for advancement of wastewater treatment plants within the context of circular economy. And that means that it is not enough anymore that they clean water, but they should also strive for water recycling, resource recovery, renewable energy generation, and of course energy reduction. And the latter is very important since it constitutes 1 to 3% of the global energy consumption, with aeration being the most energy demanding process. So how can this transition look like? It can be a shift towards different biological wastewater treatment processes, such as aerobic granular sludge that has benefits that are listed on the right because it utilizes microbial communities that are forming granules. And those can treat effluent better, so deliver better quality of water. They consume less energy because of less aeration required, and they also it also happens in one tank, so it also has a lower land footprint. But this is not always feasible because we cannot change all wastewater treatment plants. So a more feasible solution would be the addition of units that can improve the wastewater treatment process and can strive for biogas production, for example, or recirculate water back to the beginning. So there is no need for external carbon added. So the aim of this cross-study is to explore AMR profiles in conventional and advanced treatment tanks that are distinguished by the oxygen factor, making them aerobic or anaerobic, if that can be a hard discrimination. And so for this, we have in-tank ARGs and MGs data from 3 different studies: anaerobic digestion treating black water with microaeration, aerobic granular sludge treating wastewater, and a treatment line that utilizes anoxic aeration, anaerobic digestion, and partial nitrification Anammox tanks. And the data originate from metagenomics and or qPCR targeting main antibiotics groups. So from the first study, a low-cost strategy was utilized, which is microaeration, that enhances in situ hydrolysis in a single tank, and then reduces the energy consumption that is required in higher temperatures. This is a lab simulation that is targeting to treat black water, which is, has a capacity for biomethane production. And the results originate from qPCR from different oxygen inputs that range from 0 to 100 milligram per gram COD feed. And from the results of this study, we see that when methanogenesis efficiency was higher, and we see it with the orange lines on the left of the right graph, all ARGs and MGs were more abundant, with a minimum average of 2.3 times higher than in high aeration. And that was validated through statistical analysis that oxygen was correlated with the ARGs and MGs load. And also we see that the mobile genetic elements that are the blue top of the left of the graph were significantly low at the black water feed, indicating that indeed aeration may increase them in the later stages. And in literature, it is shown that actually horizontal gene transfer frequency and plasmid retention can be enhanced in low oxygen conditions. In our next study from aerobic granular sludge, samples were taken from 4 different stages, but for this cross-study, we focus on the beginning and the end of a 9-hour aeration. And we have DNA from the intracellular and extracellular DNA. And intracellular DNA originated from different size granules, large and small, and flocs. So the data here originate from metagenomics and qPCR. And metagenomics revealed that pre- and post-aeration main resistances actually remained quite the same. But it was important that the extracellular DNA resistome profiles at the beginning and end of aeration resembled the intracellular DNA of the granules. And from a taxonomic analysis, it was shown that they actually may originate from the granules. The qPCR showed no very significant load between beginning and end of aeration, but it is important to mention that at the beginning of aeration, the ARG count between the different granules and flocs was similar, while post-aeration, small granules showed higher resistance than large granules and flocs. And that may be attributed to the higher immigration capacity and shorter solid retention time that they have. And finally, our most recent study that is yet unpublished is a study where anoxic aeration, pretreatment, sludge thickening, and thermal hydrolysis pretreatment is before anaerobic digestion and partial nitrification Anammox. And From this study, we observed that antibiotic-resistant genes accumulation in anoxic and aeration did not significantly change, which is similar to the aerobic granular sludge pre- and post-aeration. But in this case, anaerobic digestion demonstrated very low ARG load, which was measured as ARGs per 16S rRNA. And these results will be validated through qPCR. And in literature, it is shown that thermal hydrolysis can actually reduce ARGs. And finally, partial nitrification Anammox surpassed all other stages. Here it is worth mentioning something that I should have mentioned before, that is an one-tank partial nitrification Anammox with intermittent aeration. And in this case, it had a high resistance load. Previous studies have shown that Anammox systems adapt very well to antibiotics by accumulating antibiotic resistance genes and excreting EPS. So maybe this could be an explanation. And from the same graph, but distinguishing the different drug categories, we see that similar to the ARGs accumulation, the resistant profiles of anoxic and aeration tanks were resembled, and also partial nitrification Anammox demonstrated high resistance to sulfonamides. And if we look at the richness of the ARGs, meaning the unique ARGs per stage, we see that anoxic and aeration profiles had the higher richness, and that may be due to their position in the system, since they are the first tanks to receive the influent. And the most unique ARGs were multi-drug beta-lactams, MLS, tetracycline, and aminoglycoside. To conclude, different AD processes demonstrated opposing ARGs abundance trends in low or no aeration, and that's possibly due to thermal hydrolysis treatment. So we know that oxygen is not the only parameter that can possibly affect AMR. Advanced partial nitrification treatment tank demonstrated significantly higher ARGs accumulation. Aeration did not significantly change the accumulation in aerobic granular sludge treatment, but the higher accumulation in small granules maybe indicate a route for dissemination. And no or low oxygen conditions potentially influence mobile genetic elements, so it is important to explore passive aeration solutions. And to finish, it is obvious that a firm conclusion on the effect of oxygen supply on the AMR load is actually not derived from these different studies, but my aim was to trigger more hypothesis tests on pollution control, because we know that this is the limitation of the oxygen supply will be the way forward, so it's important to know the effect on AMR as well. And to finish, I would like to thank my co-authors for this generous collaboration. I don't know if I have time to mention them. And it's David Calderon Frango, who performed the second study while I was a master's student, and McAnthony Norum, my supervisor, Bing Guo, David Weisbrod, Jennifer Ritchie, Yang Liu, and Mark van Lustrecht. Thank you for being here. Yeah.
Thank you very much for a great talk. I'm Jang-Woo Lee. I'm a microbial ecologist from Calgary, Canada. I have a question about probably the slide number 15 and 16, the graph, the bar plot, I guess. So at the moment, the AD there, so the y-axis is ARGs per 16S RNA. So is it, I guess, the abundances you analyze from the effluent of AD?
No, all the results are in-tank sampling.
Okay, as it is, the question probably here is that whether it is not unclear at the moment, like whether probably like AD actually reduced it or not, because there's no comparison between inflow and outflow. So have you ever taken a look at this value as well?
So you mean if we took samples from after AD was performed and not from the process?
Okay, yeah.
No, I'm asking if I understood the
Sorry?
did I understand the question correctly?
Yeah, like for instance, the inflow over outflow like this. So the ratio, whether...
No, in this study we didn't examine what is the reduction of ARGs after the process and before the process, but what will happen in the tanks, because it's not like negligible what happens in the tank. So it's not about reduction. We didn't do influent and effluent, if that's the question. We did it in the previous study, though.
Okay.
And if you're curious, there was a slight reduction in the aerobic granular sludge, yeah.
Yeah, thank you.
Hi, my name is Holly Tipper and I'm from the UK Centre for Ecology and Hydrology, which is in England. Today I'm going to be talking to you about a study that we did looking at changes to AMR during and following wastewater treatment across the UK. So we know that wastewater is an important interface between humans and the environment. And if we think about what enters wastewater, we know that it's wastewater from lots of different sources. And during the process of wastewater treatment, we get products such as bioaerosols, treated and untreated wastewater, and biosolids, and these all get applied or interact with the environment. And when we think about this in terms of the transfer of AMR and AMR-driving chemicals, we can see these sources of wastewater as sources of that and the environment as a sink. Some of the things we can learn about AMR if we look into wastewater are insights into AMR dynamics in the population, drivers of AMR dynamics during treatment, and also an indication of what might enter the environment. So it's a really important place to look. But we still have some key questions that we don't quite understand about the way that AMR behaves in wastewater, and that's generally around what happens during wastewater treatment. Some of the questions that I'm going to target in this talk include: how does AMR change during the treatment process? At what stages do these changes happen? And what are the underlying factors potentially driving changes in AMR in wastewater? So to introduce the study, this was a UK water industry research funded study. It was under their Chemicals Investigation Programme 3, so this is called SIP3, and it was looking at changes to AMR through wastewater treatment processes across the UK. And you can see here on the map, we had 10 wastewater treatment plants across England and Wales that we sampled from. And we sampled these 6 times over 12 months. So we had, every couple of months, we had a sample. The samples that we took were from influent to effluent and all stages in between. And you can see here just a schematic of one of the example wastewater treatment works that we looked at. The C samples indicate composite samples. These were 24-hour composites of liquids. And the G samples were sludge samples. The methods that we performed were molecular and chemical analysis. We did metagenomic sequencing, and we looked at AMR using ResPite, which uses CARD, and we looked at taxonomic analysis using Kraken-2. We also did qPCR of some key resistance genes. We did 16S as well, and 7 resistance genes. The analysis that we looked at for chemistry was antibiotics, nutrients, and metals. And you can see that from the 338 wastewater and sludge samples we had across this study, we got thousands and thousands of data points. So straight into the results now. This is looking at influent to effluent changes. And I'm presenting this in the form of log 2-fold changes at each site across all sampling rounds. So this is the total ARG prevalence, and this is from metagenomic data. This is resistance genes for tens of single copy genes. And you can see that across the board, at all sites, at all sampling rounds, so each of these bars is a sampling round, we're seeing reductions. And this was true of the ARG richness as well. So richness indicates the sort of amounts of types of resistance genes that we're seeing. So we saw reductions in both types of metagenomic endpoints. And if we think about the qPCR data, if I look at the qPCR data for the abundance of resistance genes, so this is just copies per litre of all genes that we targeted, we had a reduction in 97.5% of the influent-effluent pairs. So where's this happening? We saw reductions happening after secondary treatment. So this is exactly the same data as I just presented, the metagenomic ARG prevalence and richness. It's just presented a slightly different way. So you can see that the drop-off is after secondary treatment. Secondary treatment in this case was different types of activated sludge and also trickling filters. However, wastewater treatment affected different gene targets differently. This is again influent to effluent data. If we look now at one of these specific wastewater treatment plants, this is Charlton, one of our plants, this data shows metagenomic ARG prevalence, but these are for specific resistance genes. Now, if you look at the dots on top of the box plots, they are the different specific genes, and where they correspond from influent to effluent, the colour of the line and the direction of the line shows whether they increase in prevalence between those two sample types or not. So you can see that largely, most of the resistance genes that we're seeing through this metagenomic data are decreasing in prevalence, but many of them are actually increasing. And we saw this with the qPCR data as well. So this is resistance gene prevalence from qPCR gene copies per 16S RNA copies. And you can see for SORL1 and INTI1, we know they're linked. They do a very similar thing. Lots of the times they were increasing in prevalence between influent and effluent samples. Some of the sites you can see, actually it was quite site dependent, and we looked into this and it wasn't like activated sludge had different effects to trickling filters. Those sites that actually reduce in prevalence were both of those types. So it's not as clear cut as that for us. And you can see that genes like ERMB, more specific resistance genes, we found reductions across the board in all sites, all sampling points. And QNRS, it's a little bit messier. And overall, we found increases in prevalence were only seen in biocides, sulfonamide, aminoglycoside, and beta-lactam and LLSB resistance genes, and that was from the metagenomics data. So across a background of total resistance gene prevalence and richness loss, we're actually seeing increases in some specific key targets. Now, this is the metagenomic data again for influent to effluent. This is basically just a representation of the rank 1 resistance genes that we see in these different compartments. So this is using the gene ranking framework from Zhang et al., which I'm sure we all know about. You can see that influent had more types of resistance genes and also a greater prevalence, but we're still seeing some of these rank 1 resistance genes coming through in the effluent. And what's also interesting is we're still seeing a spread across the resistance categories for these genes. So what this really tells us is that wastewater, whether it's treated or not, is still an important reservoir of clinically relevant AMR. And of all the resistance genes in influent and in effluent, around 1 to 5%, depending on the site, were only found in effluent. So we're seeing, emergence of some resistance genes in those compartments that aren't actually in the influent. So now on to thinking about what might be causing these changes. This is quite preliminary analysis. I've got lots of other data to dig into to actually look at this. But what I'm going to share with you now is it's basically just the nutrient data that we have. So this you can see is a correlation plot, and I've plotted, you might not be able to see, but I've plotted things like alkalinity, pH, population equivalent, biological oxygen demand, and different measures of ammonia and nitrogen. And you can see I've also got some measures of the taxonomic changes. So I've got Chow-1 and Shannon, which are both taxonomic indices metrics. And now I'm going to overlay total ARG richness and prevalence. These are both from the metagenomic data. And you can see that we've got some strong relationships. So, the strength of the lines here indicates the strength of the relationships, and the colour of the lines, whether they're green or orange, indicates whether that was a significant relationship found. And you can see that we've got some significant, strong relationships here with some of the metrics such as biological oxygen demand and suspended solids, ammoniacal nitrogen here. And we also see strong relationships with our taxonomic indices, so Chow1 and Shannon. Now, I've had a look at, I've plotted up just a couple of these here for you. We can see here that you've got the relationships here between richness and prevalence with BOD, and we have strong positive relationships here. But interestingly, with taxonomic indices, we see the opposite. So Chow1 had a strong negative relationship with prevalence and richness, and we saw this for Shannon indices too. So this could be, indicative that we know that we lose resistance during treatment, and actually, don't show it here, but our taxonomic indices increase during treatment. So maybe we're losing resistant microbes and gaining some wastewater microbes or something similar. But this is all only correlations. Future work's going to look into causation. I also haven't presented, you might have noticed, the antibiotics data here and the metals data. It was a bit slightly messier conclusions from that. I think I could show people slides later on, but we saw that it was actually different, the correlations between antibiotics in influent and antibiotics in effluent with the resistance metrics that we have here. So I think I'm going to have to break it down further than just looking at all stages like we do here. So the key findings from this are, regardless of site and season, wastewater treatment reduces total AMR prevalence and richness, and reduction in AMR generally happened after second treatment. However, some resistant genes persisted through treatment, and final effluent can contain clinically relevant resistance genes. And reductions in total AMR prevalence and richness from the metagenomic data were associated with reductions in multiple environmental factors like BOD and measures of ammonia, and also increases in taxonomic diversity indices. So I'd like to thank the funders and thank everybody else who was on the project, and also thank you for listening.
Dominique Frigon, McGill University. I was wondering, you're showing some persistence, right? And the way you're saying it is that they came from the influent, right? The persistent genes that live with the effluent.
Yeah.
How clear are your evidence that these genes are actually coming from the influent and not just arising in the activated sludge community?
Yes, so that is an assumption, and it's not clear. And we can see that actually that's not really what's going on probably in some of the cases, because we're seeing genes come through in the effluent that we don't find in the influent. So something's happening during the treatment process that is encouraging those to emerge or increase in prevalence.
Yeah. I think this is one thing that we're increasingly seeing as well, is that the problem that we're facing is not a removal problem. The genes are removed. What we call persistence is just genes that are naturally carried by the communities that we're raising, and they're just leaving, which is a totally different problem than removing the genes coming with the influent. We can talk about more.
Yeah, thank you. Very nice talk. Eddie Citron from the Volcani Center in Israel. Sort of a follow-up on Dominique's comment. Often you see correlations between specific phylogenies and ARGs. So is it a shift in community, and those that are persisting or perhaps increasing are just due to specific populations that are able to survive in the effluents? And if you have any idea of how?
Yeah, I'm not sure whether I could pick apart which it might be. We're still yet to look properly at the taxonomic data. We've also got 16S data from this as well to look into, but we haven't linked up host with resistance genes, so I'm not sure. Genomics data. So across a background of total resistance gene prevalence and richness loss, we're actually seeing increases in some specific key targets. Now, this is the metagenomic data again for influent to effluent. This is basically just a representation of the rank 1 resistance genes that we see in these different compartments. So this is using the gene ranking framework from Zhang et al., which I'm sure we all know about. You can see that influent had more types of resistance genes and also a greater prevalence, but we're still seeing some of these rank 1 resistance genes coming through in the effluent. And what's also interesting is we're still seeing a spread across the resistance categories for these genes. So what this really tells us is that wastewater, whether it's treated or not, is still an important reservoir of clinically relevant AMR. And of all the resistance genes in influent and in effluent, around 1 to 5%, depending on the site, were only found in effluent. So we're seeing, emergence of some resistance genes in those compartments that aren't actually in the influent. So now on to thinking about what might be causing these changes. This is quite preliminary analysis. I've got lots of other data to dig into to actually look at this. But what I'm going to share with you now is it's basically just the nutrient data that we have. So this you can see is a correlation plot, and I've plotted, you might not be able to see, but I've plotted things like alkalinity, pH, population equivalent, biological oxygen demand, and different measures of ammonia and nitrogen. And you can see I've also got some measures of the taxonomic changes. So I've got Chow-1 and Shannon, which are both taxonomic indices metrics. And now I'm going to overlay total ARG richness and prevalence. These are both from the metagenomic data. And you can see that we've got some strong relationships. So, the strength of the lines here indicates the strength of the relationships, and the colour of the lines, whether they're green or orange, indicates whether that was a significant relationship found. And you can see that we've got some significant, strong relationships here with some of the metrics such as biological oxygen demand and suspended solids, ammoniacal nitrogen here. And we also see strong relationships with our taxonomic indices, so Chow-1 and Shannon. Now, I've had a look at, I've plotted up just a couple of these here for you. We can see here that you've got the relationships here between richness and prevalence with BOD, and we have strong positive relationships here. But interestingly, with taxonomic indices, we see the opposite. So Chow-1 had a strong negative relationship with prevalence and richness, and we saw this for Shannon indices too. So this could be, indicative that we know that we lose resistance during treatment, and actually don't show it here, but our taxonomic indices increase during treatment, so maybe we're losing resistant microbes and gaining some wastewater microbes or something similar. But this is all only correlations. Future work's going to look into causation. I also haven't presented, you might have noticed, the antibiotics data here and the metals data. It was a bit slightly messier conclusions from that. I think I could show people slides later on, but we saw that it was actually different, the correlations between antibiotics in influent and antibiotics in effluent with the resistance metrics that we have here. So I think I'm going to have to break it down further than just looking at all stages like we do here. So the key findings from this are, regardless of site and season, wastewater treatment reduces total AMR prevalence and richness, and reduction in AMR generally happened after second treatment. However, some resistant genes persisted through treatment, and final effluent can contain clinically relevant resistance genes. And reductions in total AMR prevalence and richness from the metagenomic data were associated with reductions in multiple environmental factors like BOD and measures of ammonia, and also increases in taxonomic diversity indices. So I'd like to thank the funders and thank everybody else who was on the project, and also thank you for listening.
Dominique Frigon, McGill University. I was wondering, you're showing some persistence, right? And the way you're saying it is that they came from the influent, right? The persistent genes that live with the effluent.
Yeah.
How clear are your evidence that these genes are actually coming from the influent and not just arising in the activated sludge community?
Yes, so that is an assumption, and it's not clear. And we can see that actually that's not really what's going on probably in some of the cases, because we're seeing genes come through in the effluent that we don't find in the influent. So something's happening during the treatment process that is encouraging those to emerge or increase in prevalence.
Yeah. I think this is one thing that we're increasingly seeing as well, is that the problem that we're facing is not a removal problem. The genes are removed. What we call persistence is just genes that are naturally carried by the communities that we're raising, and they're just leaving, which is a totally different problem than removing the genes coming with the influent. We can talk about more.
Yeah, thank you. Very nice talk. Eddie Citrin from the Volcanic Center in Israel. Sort of a follow-up on Dominique's comment. Often you see correlations between specific phylogenies and ARGs. So is it a shift in community and those that are persisting or perhaps increasing are just due to specific populations that are able to survive in the effluents? And if you have any idea of how?
Yeah, I'm not sure whether I could pick apart which it might be. We're still yet to look properly at the taxonomic data. We've also got 16S data from this as well to look into, but we haven't linked up host with resistance genes, so I'm not sure.